Okay, this is a brief review of the cell cycle, mitosis, and meiosis, but I'm going to say up front that this does not include the individual phases, prophase, metaphase, anaphase, and telophase of either, and you should definitely review those. Okay, first things first, the basic cell cycle. So all cells go through some version of this, so we'll go through this. So when we talked about the cell cycle, we were talking about most cells. So for the human body, we're talking about somatic cells, body cells. So let me pull up the pointer here. Okay, so we're talking about cells that will begin like their life here and go around this way and then head into mitosis, and then this says cytokinesis. So we're talking about going in a clockwise direction. And for meiosis, we're talking only about cells that will develop into sex cells, and that's for all reproducing organisms. So for humans, that would be called germ cells, and they become sperm and eggs. And of course, for other organisms, it's whatever the precursor to their sex cells is, but we'll focus on humans for the sake of simplicity. All right, I hope I didn't just mess up the recording, but whenever I try to go back, I end up messing things up. So sorry about that. Anyway, so if you're trying to quiz yourself on the basics of the cell cycle, G1 here is basically, so the cell has just, if it's a somatic cell, a regular body cell, say, and it just has maybe a couple organelles and a nucleus, it's going to make its cell products, whatever that is. It's proteins. It's going to grow a little bit, and then start heading into DNA replication. So remember that the main thing that cells do is whatever they do. So if it's a hair cell, it's going to make hair. So the main job of a cell is definitely going to be going on in G1, as well as making energy and making parts of the cell, and all that other regular cell stuff is definitely going to be going on in G1. And this cell is going to be growing, making proteins, and doing all that stuff. And the G does not stand for growth, it stands for gap. I don't know why, but it does. So the S stands for synthesis, and that's for DNA replication. And I will switch to an arrow to be exciting here. Okay, so woo! I always like these. Okay, so there's the arrow. Woo, DNA replication. So I pulled out the arrow because I can't emphasize it enough. I keep bringing it up, but I cannot make it as important as it should be. So, woo, this is when it happens, okay? So we're talking about one DNA becoming two, and there are 3 steps to that. And maybe you remember them, that the DNA unwinds, and DNA polymerase adds nucleotides to each side to create 2 new strands. And this is when that happens. So even though we might talk about it when we're talking about, I think we taught it during protein synthesis because that's when we got into the details of DNA. But the only reason we taught it then is because we were talking in detail about nucleic acids. But it happens during the cell cycle in the S phase. Then G2 is the cell continuing to do its regular cell stuff, but also preparing for mitosis or meiosis, whatever this cell is going to do, making more organelles so that it has plenty to do division, and then heading into the M phase, so mitosis or meiosis, and then if it's going to need a phase of cytokinesis, the actual division part after that. Okay, oops. Now I have a random arrow sitting here. I don't know how to get rid of it. It's kind of cool though. Can I move it around? Oh, I can. Cool. All right. I'm not sure why this cell cycle picture is here, but either way, you can use this right now to pause and test yourself to see if you know all the parts. All right, so try as I might, I could not make these two appear exactly the same size, but it's a common question on MCAS and other type tests to ask you to compare mitosis and meiosis. And the only reason we talk about these two together is because they're cell division. But the two really don't have anything to do with each other than that, because they're totally different processes. So meiosis is completely unique. It has to do with only sexual reproduction, only has to do with making sex cells. And if you're a female human, you are born with all the eggs you're going to have. You are done making meiosis. You only did it when you were inside the uterus of your mother. So you're all done doing it. So that excitement of your life is over. So it's a really different process than mitosis, which you have to do all of your life in a lot of your cells. So again, the only reason that these two are talked about together is because they both have a similar process of prophase, metaphase, anaphase, and telophase in them. That's the only reason we're going to talk about them together, because if you're going to take a cell and divide it, it turns out to be really handy to do this PMAT thing. But other than that, they're totally different because they have a totally different outcome. So let's talk about the outcome. Okay, let me pick a little, star. Okay. So for meiosis, it's happening in the somatic body cells. So in case you heard me saying that and didn't know what that meant, that's a really key vocabulary word because I'll say it a lot. And we are making identical daughter cells. And I don't know why they're called daughter and not son cells, but they are. And we're going to go through PMAT, and however you might remember that. I always teach Paul makes a taco because that's what I learned, and it has stuck with me for 25 years. So there you have it. And the reason, the function, the whole point of mitosis is for growth and repair. Okay, so why we go through mitosis is not to make cells. The reason we do it is for growth and repair. So because we need to do growth and repair, we make identical cells. And so that's something people mess up a lot on tests. So this whole reason part is growth and repair. And if you need to grow, you make more cells. Okay, so meiosis is a totally different reason. Okay, so we start out with these things called germ cells, which are like undifferentiated cells that are going to become sex cells. And this is just for humans because we use humans as our example for meiosis. And we are going to end up with 4 unique haploid cells. Oh yeah, for over here, for mitosis, we just end up with 2. Okay, so can I write while I'm talking? I think I can. So we're going to end up with 2 cells over here. Okay, but over here we are getting 4 unique haploid cell, meaning half. Haploid means half, so it's half the number of chromosomes. So so that when we have a sperm and an egg, they each have 23 and they come together to make the human 46. All right, so we're going to go through the PMAT twice to make that happen. All right, so we're making human gametes. And for humans, we call them sperm and eggs. Okay, so big difference. Here we have identical, and we're only making 2. Here we're making 4 unique haploid cells. All right, so here if a cell started out haploid, for example, which our cells are not haploid, but if you start out with a haploid cell, you're going to end up with a haploid cell. If you start out with a diploid cell, you're going to end up with a diploid cell. So whatever you start out with, you're going to end up with. Okay, so here's the big picture of mitosis. This is a really, really fantastic drawing on my part. Those